Now we get into the the platelets, another formed element inside of blood. So we have red blood cells, we have which are erythrocytes, white blood cells, leukocytes. White blood cells are bigger than your red blood cells. We also have these small formed elements known as platelets, which I mentioned before have to deal with blood clotting, and we'll talk about that here. Platelets. All right, so the general structure, platelets, they're small cell fragments, so generally be smaller than the red blood cells. So you have the bigger white blood cells, smaller red blood cells, and then the cell fragments, which are the platelets. They're small cell fragments surrounded by plasma membrane, right? These are the smallest of the formed elements, like I just said, smallest of between the white, the red, and the platelets. Platelets are the smallest, so remember that. They're involved in hemostasis, which is the process that stops our body from bleeding. Right. The bottom line is we have shown already that our body needs blood in the body. That blood needs to continue to circulate throughout the body. If if for whatever reason we suffer an injury and and blood starts leaking out of our body or out of our circulatory system, that becomes problematic very quickly. So hemostasis is the process, kind of very similar to that term homeostasis that you probably remember from part one of this course, but it's the process by which our body maintains blood, stops blood from leaving the injured blood vessels. Okay, anytime you cut yourself, you may you may think you're just cutting your skin, but if you cut yourself to the point that you're bleeding, you've probably cut into some smaller blood vessels. Okay, platelets don't have nuclei or organelles found in most whole cells. Okay, which is very similar to the red blood cells. Remember, they don't have the nucleus or a bunch of the organelles as well. Platelets are uh, they are developed for a specific function, and they do that function well, and they don't need a whole slew of organelles to be able to perform their function. They do have several types of granules, which we noticed from the leukocytes, granules in the cytoplasm. Remember, granulocytes are granules in the cytoplasm that that granulocytes have granules in their cytoplasm that take up that we can dye and that take a specific color. Remember those? So don't forget those from the last section. But platelets have several types of granules which contain clotting factors, enzymes, mitochondria, glycogen deposits, right? So they do have the ability to do some oxidative catabolism, some movement. We're going to see that that's important. They actually contain some actin and myosin filaments, so they have the ability to do some movement, which is going to be important in the process of hemostasis. This is the general structure of the platelet, right? Platelets, a cell fragment cut, um, fragments of cells are platelets, right? So you have some microtubules, which is some actin and myosin. Remember that actin and myosin from the muscle chapter. Mitochondria, you have some oxidative catabolism that can happen, some energy can be made, and you have granules, and granules which are embedded. Notice there is no nucleus in the platelet structure. They are not going to be going through cell division, and they're also not going to live very long. All right, so thrombopoiesis, how are platelets formed? We talked about erythropoiesis, the formation of red blood cells, leukopoiesis, formation of white blood cells. Now we have thrombopoiesis, which is the formation of your platelets. Know all three of those terms and what they refer to. Platelets begin, the HSCs differentiate into precursor cells, megakaryoblast, and just as with the red and the white blood cells, I'm not going to expect you to know every step in the making of these different cell types. I won't test you on those per se, but what I do want you to know is the general idea, right? So these megakaryoblasts develop into what is called a megakaryocyte, which goes through repeated cycles of mitosis without cytokinesis. Mitosis, cell division without cytokinesis, breaking the cell apart into two separate parts. So the cell divides, but it still stays as one cell. So basically, just gets bigger and bigger and bigger as one big cell instead of dividing and you know normal mitosis. A cell, this it doubles and then splits apart by the process of cytokinesis and forms two cells. But this cell is going to just form a one big cell. Mature megakaryocytes stimulated by hormones, thrombopoietin, uh, send cytoplasmic extensions through the clefts in the bone, marrow, sinusoids to the bloodstream, break off into thousands of platelets. All right, so we have this one cell, this megakaryocyte, goes through the process of mitosis over and over, forms this big cell, and then breaks apart, and the bunches of platelets enter into through the clefts in the bone and enter into the bloodstream via the bone, from the bone as well. Remember, red blood cells, white blood cells, uh, all are made and then enter the bloodstream as well. Platelets have a limited lifespan, about 7 to 10 days. Notice how short that is. Remember, the red blood cells lasted 100 to 120 days, and we have platelets for 7 to 10 days. After that, they're removed by the liver and the spleen. Remember, the spleen is where red blood cells also go to be removed from our bloodstream. All right, so remember to pick up on those two things. This is a diagram showing you thrombopoiesis. A cell basically just gets bigger and bigger and then breaks apart into a bunch of pieces, and then those pieces uh, eventually form a cytoplasmic arm through the bone. Remember, this is happening in the bone marrow, through the bone, and then enters the bloodstream. Right, and then here we just have a diagram putting it all together of how the different hematopoietic stem cells, the uh, HSC hematopoietic stem cells, leads to your different types of formed elements that are in blood, your erythrocytes, your platelets, your different leukocytes. Remember, there's the granulocytes and the agranulocytes, the neutrophils, the eosinophils, the basophils, the monocytes, and the T and B cells, which are the lymphocytes, right? So these are how all these are formed. Just understand the general idea of this chart and the major differences between these different types of cells. Know which ones are red blood cells, white blood cells, platelets. Know which ones have nucleuses, which ones don't have nucleuses. Know which ones have granulocytes, which don't have granulocytes. Know similarities between notice the similarities between certain cells. For instance, the platelets and the erythrocytes don't have a nucleus. Know how the general lifespan of these different cells, where they go to be removed from the body or where they go to die per se. Know all that information, right? And now let's go into the next part, which is hemostasis, which we're going to go into more detail about. All right, so we have hemostasis is, as I stated, it is this process by which our body basically prevents blood from leaving the leaving circulation, leaving the body, leaving the blood vessels in a way that we don't want it to leave. Um, we want blood to stay in our body. We want blood to stay in the blood vessels for the most part. There are ways that blood can get out of blood vessels, but in a right way. But for the most part, we want blood to stay in the blood vessels, stay in the body, continue circulating through the arteries, arterioles, capillaries, venules, veins, through the heart. We want the blood to continue its circulation until those blood cells are no longer useful to us. But the process of hemostasis is very simply to form a gelatinous blood clot, uh, which plugs broken vessels, right? So the whole idea is there's been an injury, somehow an injury has occurred at a, to a blood vessel. It could be, you cut yourself. It could be you banged your banged a part of your body against something hard. That can break a blood vessel. You can have bleeding happen. You see a bruise or something. Blood vessels can be broken. Many different ways this can happen. But whenever it does happen, your body does what it is designed to do, and that is to prevent the loss of blood. Right. So these are the steps: vascular spasm, platelet plug formation, coagulation, clot retraction, and thrombolysis. All right, so part one, vascular spasms. So a blood vessel is broken. A blood vessel is injured. Blood starts spilling out into this extracellular fluid, into the space outside of the blood vessel. Uh, the first step that happens is vasoconstriction. Vaso, your vasculature starts to constrict. Vasoconstriction. Increased tissue pressure both act to decrease blood vessel diameter. Blood loss is minimized as both blood pressure and blood flow are reduced, right? So constrict, you constrict. Take for instance, you take a water hose and you bend it. The more you bend it, sort of constrict it, if you take that water hose, the less water that's going to come out of the end. So if I have, if let's say this, my arm's like a water hose. If I have blood leaking out here and I bend it here, per se, if I bend the water hose here, the more I bend it, constrict it here, the less is going to leak out up here. That's sort of the idea. You want to constrict the blood vessel so that less blood is able to get through the blood vessel, less blood is able to eventually leak from the blood vessel. So blood flow is reduced and blood pressure below the blood pressure down the line from where it's restricting, less blood pressure. And so you have less blood that is going to be pumped out of the blood vessel where the injury has occurred. All right, so vasoconstriction. This is a this is a um, showing you the first step, vascular spasms, which lead to vascular constriction. So these spasms cause the blood vessels to constrict and less blood leaks out. So you have reduction in blood flow, reduction in blood pressure. And we're going to talk about how blood flow and blood pressure are related, very much related. But as you can see, and think of a water hose. If you start to bend the hose somewhere along the hose, less less pressure down the hose from the bend. There's going to be less pressure on that water. It's going to come out slower, and less water is going to come out. All right. Step two. And this is where things start to get a little bit tricky. All right, so see if I can fix my face somewhere. Step Step two is you have platelet plug formation. All right, so the idea here is we're sort of gonna begin to, we want to slow down the blood flow. We have a hole in the blood vessel or some kind of injury to the blood vessel. So we're going to slow down the blood flow. We're going to stop it up a little bit at first and then put a better stop into it. And then until it heals and eventually remove all of that and get back to normal. But the goal of this whole process, remember, is that we want as little blood loss as possible. All right, so with part two, we have a patch consisting mostly of platelets that adheres to the injury site. All right, so we've reduced the amount of blood flow that is going to that area that where the injury has occurred. So less blood is leaking out. Then we have platelets, which are our formed element, which we just talked about. We have uh, these are the factors, these are the steps that are involved, and you'll see this von Willebrand factor, which is a glycoprotein that binds to receptors on surfaces of platelets, plasma membranes. These are released by the injured endothelial cells. And when we talk about the structure of the blood vessels, we're going to talk about the endothelial cells that line blood vessels. So these endothelial cells have been displaced, broken apart, injured in some way. They release this von Willebrand factor that binds to receptors on the surface of platelets and plasma membranes, the plasma membranes of the platelets. This VWF, this von Willebrand factor, makes platelets sticky. They adhere to one another and surrounding vessel walls. So these platelets start to form a plug, right? Just like a plug is if you put it in your bathtub. They start to form a plug right where the blood is leaking out, right where the injury has happened, because it's the injured cells that release the von Willebrand factor. So the platelets go to the spot and remember the platelets are circulating in the blood, but the platelets go to that spot because that's where the injured cells are releasing the von Willebrand factor. These activated platelets release granules containing ATP, ADP, serotonin, calcium, clotting factors. Uh, these granule contents attract and activate nearby platelets. So the nearby platelets, not only are platelets sticking to that area where the injury happens, then they activate nearby platelets and they cause them to clump up together. And this forms a plug, which seals the injured vessel temporarily. So you see here the you'll see here the endothelium. That's the outside of that's the surface of the structure of the blood vessel. Releases this von Willebrand factor, binds to platelets, which bind to that area and attract other platelets. They get sticky and clog that area, and now blood can't get out of that broken area. Step three, coagulation. Okay, so now we have platelets stopping the blood flow from leaving that specific injured area. Now we have coagulation, a process that forms molecular glue, binds platelets, endothelial cells, and other formed elements together. All right, so we have this coagulation, this forming things
This
coming
happens
together.
by intrinsic and extrinsic factors. Don't be confused by intrinsic and extrinsic. Intrinsic simply means that it factors from inside the blood vessel, extrinsic factors from outside the blood vessel, but they both just lead to this. The key thing is fibrin. It's thread-like protein that converts soft liquid platelet plug into more substantial solid mass. So with the platelets, remember the platelets are just simply pieces of that megakaryote, pieces of a cell that are broken apart that float around in the blood, and they form this sort of loose, sort of light clog. It's like, if I guess it would be if you took like a, like a cotton ball and clogged something. Now we want to use fibrin, this thread-like protein, because we want to get back to where we used to be. We want to get back to the structure that we sort of similar to the structure, and we haven't talked about this in a lot of detail yet, but we want that artery or that vein to get back to the sort of the same structure that it had before it got injured, right? So we need something a little bit stronger. And so we have what we call fibrin, which is this protein that helps convert that platelet plug into a more solid, substantial solid mass. Okay, that's what we want. All right, so fibrin is circulating in the plasma and in platelets, and inactive fibrinogen. The fibrinogen is converted into fibrin by a series of reactions, and this event just depends on the certain clotting factors that are involved. And I don't need you to know all the clotting factors, but just know that there's two different types of pathways. There's an intrinsic and extrinsic pathway that leads to this fibrin, which is going to help strengthen this plug. And that's really what I'm hoping that you know. So we have the intrinsic pathway. So as I said before, these are clotting factors that are found in your blood. So they help us get to the fibrin, help us do the clotting. These are factors found in the blood, and you can see them here. And then you also, and so you can see the diagram here. This gets us to that fibrin with clotting factors found in the blood, but you also have extrinsic pathways. These are initiated by factors outside of the blood. And then, so these are both where, so factors, clotting factors in the blood and clotting factors outside of the blood are both working together to strengthen this clog, strengthen this sort of this plug, which is originally just a platelet plug, strengthen this platelet plug with this fibrin. Okay. And so you're getting strength from the outside as well. So your body's working on both sides of the injured blood vessel to coagulate it, strengthen it, and to make sure that blood does no longer leaks out. Right, and this is what it looks like after it's all coagulated. And then the next step in the process is clot retraction. Right, so occurs as coagulation, cascade nears its completion. So now we have these actin and myosin fibers. Remember, platelets have these actin and myosin fibers. Remember, platelets also have mitochondria, and we have platelets that are were the initial step in the formation. What, not the initial step. After the vasoconstriction, you have this platelet plug. So the platelets were the initial piece of this clog, this blood clot. So there's still platelets involved, but now the actin and myosin fibers involved in the platelets contract and bring the edges of the wounded vessel together. So now we start bringing the edges of this wounded blood vessel together. Why do we want to do that? Because we want to get back to where, ultimately to where it used to be. Serum, you have a fluid consisting of plasma without clotting proteins forced out of clot during clot retraction, right? So you have serum that's released, actin and myosin fibers bringing the edges of that broken blood vessel back together. Oops. So that's called clot retraction. Start bringing that structure back together. Start healing. And then the last step is thrombolysis, right? We don't want that blood clot to stay there forever. That ball of platelets, that clog, that fibrin, we don't want that to stay there together. After it starts to heal and come back together, Now we no longer need that blood clot. So what happens? We want to break it down. Thrombolysis begins after the injury has healed and blood clotting is no longer necessary. Remember, what made it strong was this fibrin. So you have this fibrinolysis, which breaks down the fibrin glue, that fibrin protein. Endothelial cells release this TPA, initiates thrombolysis. We break it all down. The point that I have here at the bottom is when I want you to remember the plasmin degrades fibrin and clot dissolves. Remaining components of clot disassociate from endothelium. That's the surface of the blood vessel. They disassociate from the endothelium and the process is complete. So you want to break everything back down into its pieces and parts, and have it all nicely dissolve. That's the goal, right? So that's your thrombolysis. So we form the clot. So the blood vessel breaks. We can vasoconstrict to try to reduce the amount of blood that is flowing out of this broken blood vessel. We then have platelets rush to the site. Those platelets get attract other platelets, stick to the site, form a loose plug. Then the fibrin gets what's the word? I'm losing my words. The fibrin gets activated. The fibrinogen gets activated, forms fibrin. You form this strong structure. The actin and myosin in the platelets starts to bring the edges of that broken blood vessel together. Eventually it heals up, and now we no longer need that blood clot. We have thrombolysis. We break everything apart. It dissolves, and everything goes back to normal. Now, of course, this can go wrong, and if it doesn't dissolve, you would have blood clots in your bloodstream, which can happen if that's a problem. Clearly, that's a problem, because then that could block blood from flowing in different parts of your body. This is just a slide that shows the big picture of this whole process. And so you should go through this and definitely go over it and figure out, and understand what's going on. The different clotting, there were different regulations of blood clotting and how they play a different things that play a role in blood clotting. Fibrinogen is going to be one of the main things at the top of the list. So you'll definitely see that. So just know that one. Also know prothrombin and tissue factor. Go through the list, but those three definitely you want to remember. All right, so clotting is regulated by a positive feedback mechanism. It must be tightly regulated to prevent mishaps. What are those mishaps? Well, like I said, you could have blood clots forming where they're not supposed to form, blood clots not dissolving, floating through your bloodstream. That would not be good. They could block the flow of blood. Blocking the flow of blood means you're blocking oxygen and nutrients potentially to different parts of your body. So blood clots where they're not needed could be very problematic. So endothelial cells produce and secrete two chemicals that regulate first and second stages of clot formation. Prostacyclin inhibits platelet aggregation. Nitric oxide causes vasodilation. Remember, vasoconstriction happens when there's a problem. So we want to vasodilation get back to normal. Endothelial cells and hepatocytes produce anticoagulants, which inhibit coagulation, right? And there's three listed here: anthrombin 3, heparin sulfate, and protein C. And we'll talk you'll see heparin sulfate. Some of you who are in the medical field have probably heard of heparin. So we'll talk about that in a little bit, but these are anticoagulants. So these are both things that are important, that we want to understand. Sort of, they're important for the regulation of blood clotting. All right, this is just a chart that sort of points out regulators of clotting, blood clotting. You can have clotting disorders, a condition in which clotting is not regulated properly. And as I said, it can cause some major issues. I mentioned blood clots that are happening that are not supposed to happen, but there also is an issue of blood clots don't happen. I actually have a cousin, my cousin's wife actually passed away due to a blood clot issue. She bled out, and just from a small cut on her hand, because she has an issue. She had an issue when she was alive. She had an issue with blood clotting. Her blood did not clot properly, so it would not clot, and so it was very dangerous for her to have any type of cut or any type of injury, because her blood did not clot properly. Therefore, she would bleed profusely from even the smallest injury. So I'm very familiar with this, actually. All right, so either direction, it can cause problems. Bleeding disorders, increased blood loss from even minor injuries, which I was just talking about. Hemophilia A caused by a shortage of factor 8, hemophilia B, shortage of factor 9. These are two blood clotting factors. If they're not available, you can have your blood doesn't clot properly. So therefore, because it doesn't clot, you bleed, you lose more blood. You also have in the other direction, so hemophilia will be your blood doesn't clot when it needs to. The other issue is hyper, too much, coagulable. Coagulate means to come together. So hypercoagulable, your blood clots too much. Results in a formation of inappropriate clots, which is called thrombosis. You'll skip down to where it says deep vein thrombosis. I think that one and pulmonary embolism are the two that you'll see most commonly, or you probably have already heard of. So deep vein thrombosis, thrombi commonly form in deep veins of legs. So you'll see this especially in older people, but it can happen in younger people as well, where they have blood clots that are forming in their veins in their legs, and this can be very problematic. Actually, my mother-in-law had a deep vein thrombosis, and they had to filter her blood to remove some of the blood clots. And one of the dangers of that is pulmonary embolism, where if you have deep vein thrombosis and if that blood clot circulates through your system and gets into your lungs and can block the blood vessels in your lungs, now oxygen doesn't get through your body. And so that can be a huge issue. If you're blocked, at your lungs is where your blood vessels meet your lungs. That's where your blood vessels transfer oxygen into your body, transfer carbon dioxide out of your body. So if you start getting into those blood vessels and blocking those blood vessels, now the oxygen that you breathe in, the carbon dioxide that you breathe out, can't get to where it's supposed to go. And now parts of your body, and now your body's going to really have some major issues. And so these are just some problems that can happen simply from your blood clotting improperly. You have medications that you can take, which are listed here. Anticoagulants, heparin, heparin. These are used often in the hospital. So if you're working in a hospital, you may want to become familiar with this as an anticoagulant. Breaks up blood clots. They give it to people for different things that could potentially cause blood clots. Aspirin, antiplatelet drugs, inhibits enzymes that contribute to aggregation of your platelets, so your platelets don't clump together. Aspirin can help with that.